Bioinfocongress IV Associate Professor Dr. Zeynep Baharoğlu Epigenetic Adaptation to Antibiotics in Gram-Negative Bacteria. After graduating from Pierre High School in Istanbul, Dr. Zeynep Baharoğlu studied cell biology and microbial genetics at University Grenoble and University Paris Cité, respectively, in 2000. She spent her last undergraduate year at the University of California, Irvine, as part of an exchange program. She received her PhD from Sorbonne University in 2008 for her research on E. coli DNA replication and repair mechanisms in the CNRS. In 2008, she joined the bacterial genome plasticity team at Institut Pasteur as a postdoc. As a researcher at Institut Pasteur since 2014 and group leader since 2020, she has focused on the response of gram-negative bacteria to low-dose antibiotics and more recently on the impact of RNA modifications.
Dear Dr. Zeynep Baharoğlu and participants, welcome all of you to our Bioinfocongress IV event. Dr. Zeynep Baharoğlu will give us a talk today on epigenetic adaptation to antibiotics in gram-negative bacteria. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Zeynep Baharoğlu, welcome back.
Thank you.
If you wish, you can start your presentation.
Okay, thank you very much for the introduction. So I would like to thank the organizers for having invited me. It's an honor to be here, so I'm very happy to be here today. So thank you very much. As you said, I will present you today my research topic about antibiotic resistance and tolerance and involvement of epigenetic modifications of RNA. So I just would like to mention that a few words just about the earthquake that happened. So my family is from Iskenderun, and so I would like to say that my thoughts are with everyone who suffered from this, and I just wanted to share a nice photo to remember it, nice things, and to be to have some hope for the future. So okay, so now I'm at the Institut Pasteur in Paris, France. So my group is studying bacterial responses to stress in the presence of antibiotics. So antibiotic stress is, of course, very important nowadays because of antibiotic resistance development. Because bacteria are becoming more and more antibiotic resistant. So in the right here, you can see the years of introduction of several antibiotics here in the market, and the years where resistant bacteria appeared. For example, here for tetracycline, you can see that tetracycline was introduced in the 50s, and in 1959 there were resistant bacteria which appeared, but now this happens even more frequently and in a shorter time. For example, here you need just for ceftaroline, it took only 1 year for resistant bacteria to appear, so it's really a major problem. So there is also tolerance to treatment for sensitive bacteria. I will talk about this a bit more in the next slides. But there can also be relapsing infections of susceptible bacteria, which are not resistant, but which still tolerate antibiotic treatment. And so it's a major public health issue. So in the lab, we are more concentrated in understanding the effect of antibiotic stress at antibiotic concentrations below the lethal concentrations. So we use antibiotics at doses that do not kill bacteria, which we call submic, for subminimal inhibitory concentration. And these kind of doses can be found in the environment because of industry waste, healthcare waste, or even farming and animal husbandry, where the waste containing antibiotics goes in the soil and in the aquatic environment at quite high doses, which are still below lethal doses for bacteria. And the bacteria can encounter these low concentrations of antibiotics in their environments. And these concentrations do not kill bacteria, of course, but they are stressing agents, and bacteria have to develop strategies to grow well in presence of this kind of stress. So it has been shown by different labs that these kind of submic antibiotics are stressors, and bacteria respond to these stresses. And there is a question about the fact that antibiotics grow in presence of low doses of antibiotics, and whether this can lead to antibiotic tolerance and resistance. So just a definition slide of words that I will be using in my presentation. So these are mostly about resistance and tolerance. So what is resistance? Bacteria are called resistant when when we need more antibiotics to kill them. So here you have the susceptible bacteria, where the MIC, minimal inhibitory concentration, is 2 here. And if you have a resistant clone, if resistant strain of bacteria, you need more antibiotics to kill it. The MIC is increased here to 62 in the example. So this is resistant. Unlike that, tolerance is a bit different. So tolerance does not change the MIC. So if you have a susceptible population or a tolerant population, their MIC is the same. So you need the same dose of antibiotic to kill all of the population. But the difference is that if you do a killing curve, so you take bacteria, you treat them with the antibiotics, and then you see the proportion of survival. You see that susceptible ones in green here die faster than tolerant ones. So here, for example, when you look at 6 hours of treatment, you still have some tolerant bacteria which have survived the treatment, while susceptible bacteria were cleared. So tolerant populations usually do not resist to higher doses, but die more slowly. And this is usually also, it can be measured in competition experiments. For example, in the lab, if you mix 2 populations at equal levels, and then you at equal proportions, and then you grow them for a number of hours, and then you measure which bacteria is present in the culture. You can see that one has invaded the culture, and it's the tolerant one usually, or the one that grows better, which is the tolerant one. So in the lab, we use as model the cholera pathogen Vibrio cholerae, which is a gram-negative bacteria. And we study its response to submic antibiotics, and we asked whether we could find some genes that are important for growth and response of Vibrio cholerae to submic antibiotic stress. So to do that, we take the Vibrio cholerae genome, and we use a transposon, which is called Mariner transposon, which can insert in the genome in a random way. So we build large libraries of Mariner transposon insertion libraries into Vibrio cholerae genome. And in these libraries, theoretically, we have for each gene at least one insertion in this gene. And when the transposon gets inserted in a gene, usually the gene is inactivated because there is some random chunk of DNA inside. So then we take these libraries and we grow them either without antibiotics or with different antibiotics at concentrations which do not kill bacteria. So we took half of the MIC. And we grow them for 16 generations, which is not very long for bacteria. If you're a microbiologist, you would know it's like an overnight culture. So it's not a very long evolution. And then we sequence the regions flanking the transposon, which gives us the locus where the transposon was inserted. So it means that this gene is inactivated. So for example, here after 16 generations without antibiotics, we have gene A, gene B, and gene C, in which we detect insertions in blue here. And if we look with antibiotics to the right, we see that in gene B, we have lost detection of insertions. It means that when there were insertions in gene B, when gene B is inactivated, the bacteria containing these insertions were lost. They died. So they couldn't survive the stress, the antibiotic stress. So by comparative analysis of sequences with antibiotics and without antibiotics, we can infer the genes which were important for growth in the presence of this antibiotic. It is not depicted here, but we can also have the opposite. So for example, we can have a gene C, for example, where the number of insertions would increase in the presence of antibiotics. And that would mean that the inactivation of this gene is beneficial and bacteria with insertions in this gene grow better and invade the population. So we did that with two different antibiotics because they have different targets. So tobramycin, which targets the ribosome, and ciprofloxacin, which targets the DNA. And then we analyzed all the sequences that we have, and we end up with genes in which we have increased insertions or decreased insertions. So here I show volcano plots. So in the x-axis, to the right, it's the number of insertions which increase. And to the left, it's the number of insertions which decrease. And the y-axis is the inverse p-value, which means that the higher the gene is, the more significant is the result. So what we see here is that, for example, we see DUS-B here in the right side of the plot. It means that when we have insertions in this gene, DUS-B, the bacteria grow better in presence of tobramycin, which means that deletion of DUS-B is beneficial for growing in tobramycin. And to the other side, we have TGT here. When you inactivate TGT, bacteria get lost. It means that TGT is important for growth in the presence of antibiotic. And we did the same thing with ciprofloxacin. And in both cases, we identified many genes associated to RNA modifications. So tRNA modifications and ribosomal RNA modifications. So I will explain this more in the coming slides. But when we do gene ontology enrichment analysis of these genes, you can do it online. So you have a list of genes that have different levels of insertion. You take these genes and you see if there are significant changes in gene ontology groups classified by function. So for example, here you can see that with the red arrows, we can see that RNA modification genes appear in a significant way in this list. So it means that it's not just that we are seeing more RNA modification genes, it's that it's statistically significant. Okay, so before I go into RNA modifications, I would like just to show again the central dogma that many of you know, of course. The central dogma is that we have DNA. The DNA gets transcribed into mRNA, messenger RNA, which gets translated by the ribosome to proteins. And so genetic mutations can happen in the DNA. They are hereditary, so they can be transmitted from generation to generation. And there is also epigenetic modifications. And for example, when we hear about epigenetic modifications, usually it's DNA methylation that we talk about. And so DNA can get methylated. And this is not genetic, it's not a mutation, but it's an epigenetic modification of DNA. And DNA methylation in bacteria was all associated with virulence, antibiotic resistance, cell cycle regulation, etc. But there is also modifications in RNA. So the messenger RNA can be modified. This is rare in bacteria. In eukaryotes, it's much more present. And other RNAs can also be modified, which are transfer RNAs and ribosomal RNAs, which compose the ribosome. So it can be methylations, but there are also other modifications. And so this kind of modifications in bacteria were very few phenotypes were described. Their role is quite not very well known. So as I said, there are different RNAs inside the cells. So inside bacterial cells, you have messenger RNAs, transfer RNAs, which bring amino acids for transcription to the ribosome, ribosomal RNAs inside the ribosomes, and some non-coding RNAs. So at least 3 of these RNA types are involved in translation, protein synthesis. So we could imagine that if RNA modifications are present or absent, that could impact tRNAs, mRNAs, and rRNAs, so protein synthesis. This is just to show you, to give you an idea where modifications can be. So this is ribosomal RNA on a 2D depiction of ribosomal RNA. So this is the 16S ribosomal RNA. This is the 23S ribosomal RNA. And each colored square or round or triangle are different modifications that are present in ribosomal RNA. In E. coli, they are well described. So they are mostly involved in the stability of the ribosome or the structuration of the ribosomal RNA, but for some, the function is not really well known. For tRNAs, so this is a tRNA. This is the anticodon of the tRNA, which binds to the codon, and here you have the amino acid that is attached to deliver the amino acid for translation elongation. So in E. coli, there were almost 80 modifications that were described for tRNAs. So many of these modifications happen at the anticodon loop. So here in the positions 34, 35, 36, 37. But you have also other modifications elsewhere on the tRNA. And what's interesting is that when you delete genes which do these modifications, usually you have no phenotypes, no growth phenotypes. So bacteria in the lab continue growing well like the wild type. They have no growth defects, except for 3 of them. So that was interesting because it could mean that these genes are not important during growth in rich media in the lab, but maybe they have some function in another situation or in response to stress. And since we, in our screen with antibiotics, we identified many of these genes. They are here in red square. All these genes, we have identified them in our screen as important during growth in the presence of tobramycin. So what we did is that we deleted each gene. So we did single deletion mutants in Vibrio cholerae of each gene. And we did some competition experiments. So we mixed the wild type and the mutant strain at equal proportions. We grow them together and then we plate and we see if one of the two strains wins or if they are still at equal proportions at the end. So this is presented here in these graphs. It's the competitive index in a log scale. So 0 means that they are still at equal proportions. There is none of the strain wins. And here, as you can see, it's the non-treated condition. So we just grow them in rich media. And as you can see, the majority of the mutants are at 0. It means that after the competition, they are like the wild type. They have no growth defect or they don't grow faster, except for a few of them which grow slower and which get lost. But then we did the same thing with the antibiotics in the presence of aminoglycosides, ciprofloxacin, trimethoprim, carbapenicillin. So these are 4 families of antibiotics which target different targets in the cell. So carbapenicillin is the membrane, trimethoprim is the DNTP pool, ciprofloxacin is the DNA, and aminoglycosides is the ribosome. And here you can see that we have differences now. For example, here the TGT, it gets lost from the culture. So you have almost 100 to 1,000-fold less delta TGT strain than the wild type at the end of the competition. It means that it has a really large growth defect in the presence of tobramycin, which is not the case, for example, in the presence of Cipro or the other antibiotics. We can also have another case where the mutant wins. Here, when you delete this gene, the mutant strain wins, which means that the deletion of this gene is beneficial. And it's not the case for other antibiotics. And for each antibiotic, we have some different genes that have an effect. For example, here for trimethoprim, it's this one. So doing that, we tested 25 tRNA or RNA modification mutants. And what we see is that we confirm that what was known in the literature, that when you delete them, there is no growth phenotype in the absence of antibiotics usually. But in the presence of antibiotics, we have sometimes strong growth defects or benefits. And this is dependent on the antibiotics or the gene, so it's a specific response. It's not like you delete all the genes and they all become slow growers and they all lose. It's not like that. It's really specific. So then we decided to see how this can happen. So we decided to characterize more one of the deletion strains, which was the TGT. So TGT makes a modification called Q-zine on some tRNAs. And it was mostly studied in eukaryotes. So in mammalian cells, this modification is associated to cancer, cell proliferation and malignancy, and also to oxidative stress responses. In yeast as well, it was associated to oxidative stress because it influences mitochondrial protein expression. But in bacteria, there is almost no study linking this Q-zine modification with any specific phenotype. So now we have one strong phenotype and we decided to study it. So as I said, to show it again here, this is a competition experiment. NT means non-treated, so without any antibiotics, both strains are equal, but when we put the tobramycin in it, the TGT loses, delta TGT. We also tested this with high doses of tobramycin, lethal doses, so 5 times more than the minimal inhibitory concentration here. So we grow bacteria, then we take them and we treat them with this dose of antibiotic, and we plate them at different time points to see the survival rate. And we see here in the blue that the wild type dies at this rate that we can see, and the delta TGT mutant deleted for this modification dies faster here. So it is less tolerant to tobramycin. So TGT modifies 4 tRNAs in the cell, which are tyrosine, aspartate, histidine, and asparagine. So we wondered whether one of these amino acids were more important in the response to tobramycin. So just to show you how modification of tRNA could influence codon decoding, this scheme, so it shows again the central dogma, DNA, messenger RNA, and protein. So here I put the example of tyrosine codon. So tyrosine has can be coded by 2 codons, TAT or TAC. So when this is transcribed into messenger RNA, it becomes TAT becomes UAU and TAC becomes UAC. So this UAC codon has a tRNA anticodon which can recognize it, which is AUG, which forms a Watson-Crick base pair here. And so, depicted here, so this is the expected situation. But when you have a cuisine modification here at the G becomes a Q, and then here we don't really know what happens, if this is more stable or less stable than this, for example. So for the other codon, TAT, as I said, messenger RNA is UAU, and you would expect a tRNA which is AUA, but this tRNA does not is not present in the genome in gram-negative bacteria. So they do not have this tRNA AUA. So this codon has to be decoded by the same tRNA decoding the TAC codon. So what we have is depicted here again. So the UAU is decoded by the AUG. And here you have a mispair. But this is the real life situation. And when you have a cuisine instead of a G, again, we don't know what happens if this affects the stability of the codon decoding and codon recognition. So here we can see that modification of the anticodon of a tRNA can impact the codon decoding and recognition in general. So this happens for the tyrosine, but also for the other tRNAs that are modified by cuisine. So what we did is that we constructed a fluorescent reporter, GFP, green fluorescent protein, where we introduced some codons at the beginning of the gene. So we wanted to test the four amino acids for which tRNAs are modified by cuisine, and these are all the 8 codons here that I show. It's not very important, but we constructed reporters where we put repeats of the codon that we want to test, and if the codon is correctly decoded, the green fluorescent protein will be synthesized and it will be fluorescent. But if there are problems for decoding the inserted codon, then the fluorescence will go down because there will be less protein GFP made. And so what we saw is that only for one codon we had a difference between the wild type and the non-modified delta TGT strain, and it's the TAT codon, the tyrosine TAT codon. In the delta TGT mutant, it was more fluorescent. It means that more proteins are made when the modification isn't there at the TAT codon. So this suggested that there is more efficient amino acid incorporation in the absence of modification at tyrosine TAT. So we constructed another reporter, which is a bit different. So it's a beta-lactamase gene which codes for carbanicillin resistance. It's quite well described, and we know that there is a tyrosine in it that is important for its function. So if you remove this tyrosine, the cells become sensitive to carbanicillin, even if they have this gene. So the beta-lactamase gene, this tyrosine codon is a TAC, and we made a synonymous mutation. So we changed the TAC to TAT, which is still a tyrosine. So both versions of this gene code for the same protein, okay, but it's just not the same codon. So we introduced these two versions in wild type and in delta TGT strain. So in wild type, both grow at the same rate in the presence of carbanicillin, but in delta TGT, one grows faster, and it's the TAT version. So even when we have two genes coding for the two same proteins, the codon in it is very important for the expression of this gene. So in the absence of modification in delta TGT, when you have a TAT here, you have more beta-lactamase protein that are made, probably. So these experiments showed that the TAT codon is better decoded in the absence of cuisine, but then we would like to know what's the link between this and the fact that delta TGT is sensitive to tobramycin. So to ask this question, we did some proteomics. So we took proteins from, we grow the cells in the presence of tobramycin, and then we quantify proteins from the wild type and proteins from the delta TGT, and we compare the abundance of all the proteins in mass spectrometry. And what we saw is that there was one protein which was really much more abundant in delta TGT than in wild type, which is RS6A here. There were 13 times more RS6A in delta TGT, which is a lot in this experiment, in mass spec proteomics experiments. 13 times, it's really a lot. So we wondered if this high abundance of RS6A could explain first the sensitivity to tobramycin. So to understand that, we just expressed this protein RS6A in the wild type, so we overexpressed it. And when we overexpress this protein, we see that the growth in tobramycin is decreased. So the overexpression of RS6A can explain why TGT is more sensitive to tobramycin. Okay, so what is RS6A? Briefly, RS6A is an inhibitor of oxidative stress response. So we have this is not really important now, but I will just to explain. We have SOX-R oxidative stress response regulator, which is active when there is oxidative stress, but when we have more RS6A, protein, it inactivates SOX-R and the oxidative stress response. So if you have oxidative stress and too much RA6A, then the oxidative stress response of the bacteria is diminished. And what we think is that maybe in delta TGT, since we have more RA6A, then maybe we inactivate the oxidative stress response, and maybe that's why the top sensitivity happens. We thought that because we know that in the presence of tobramycin, there is some oxidative stress in the cells. We had already observed that previously. So, as I said, if you overexpress RS6A, even in the wild type, it grows much more slowly in the presence of tobramycin. And what was interesting in this protein is that it has 6 tyrosine codons, and 5 of these 6 tyrosine codons are TAT. So in the Vibrio or gram-negative bacteria genome, if you randomly take any protein from the genome, if it has 10 tyrosine codons, 5 are TAT and 5 are TAC. Usually it's 50%, 50-50, as shown here. But for RS6A, you have 80% of TAT, which is called a codon usage bias. So RS6A has a bias toward TAT codons. So what we did, we thought maybe RS6A is much more expressed in delta TGT because of these TAT codons. So we replaced these TAT codons by TAC codons, which are also tyrosine codons, and they make the same protein. And we asked if this TAC version is expressed at high levels like the TAT version. And so this we checked that using fusions again with the green fluorescent protein. So we measure fluorescence here. So with the TAT version, we confirm that in delta TGT we have much more RS6A. But when we replace the TAT codons with TACs, we lose that. So we don't have this high expression level anymore. So it means that RS6A is highly expressed in delta TGT because it has more TAT codons than TAC. So this brings me to the concept of modification tunable transcripts, which were defined in eukaryotes first in this paper here as transcripts, so mRNAs, which use specific codon biases, so which do not have the same codon usage as the rest of the proteins in the genome, which encode stress response proteins, and for which translation is influenced by 3NA modifications. So in eukaryotes, usually they are involved in cell cycle progression and cellular stress. So we propose that RS6A can be qualified as a modification tunable transcript because it has a codon usage bias. It has more TAT than TAC codons. It encodes a stress response protein because it regulates the oxidative stress regulator, and its translation is influenced by the chiosine modification, TGT. But for this to work, you need to see if the modification levels of inside the cell can be dynamic, if they can vary. Because if they do not vary, you cannot. They cannot be a stress response. They cannot function as a stress response, because when there is a stress, the modifications have to go up or down so that the cell responds to that. So we wanted to see whether there are conditions where the TGT gene is more or less expressed. And we found that yes, some conditions. So here we measure mRNA levels of TGT, and we saw that in presence of tobramycin or in other conditions, there are some conditions that upregulate TGT. And then we wanted to see whether upregulation of TGT is associated to more chiosine in tRNAs. So we looked at chiosine levels in tRNAs by mass spectrometry, and we see that in this condition where TGT is highly expressed, we also have more chiosine in tRNAs. So this shows that chiosine modification levels can be dynamic, and maybe they can be used by the cell to respond to antibiotic stress or other stresses, maybe oxidative stress. So what we propose here as a model is that when there is a stress, so in our case it's antibiotic stress, but it can be other, starvation, temperature, acidity, RNA modification enzymes can be activated or inactivated. This also can happen. So in our case, it was TGT. This leads to changes in modification levels. So maybe we have more chiosine in some tRNAs. And this impacts protein synthesis on specific proteins. So you have specific proteins that are up or down regulated, which creates some heterogeneity in the phenotype, because even if the genome is the same, the expression levels of some proteins change. And this leads to adaptive response to stress and leads to homeostasis. So then we wondered, so we found RS6A protein in a proteomics experiment, but we wondered whether we could look in silico for gene sequences to find some candidate genes which could be regulated by the same modification mechanism. So in Vibrio cholerae, we looked at genes which have more TATs. And one category here was interesting, it's DNA repair. So DNA repair genes are shown here in this heat map. And you can see that, so in the left, it's the TAT codon, and when it's red, it means there are more enriched. And in the left, it's the TAC codon, and you can see that the rec genes, rec A, rec O, rec R, which are involved in DNA repair, are enriched in TAT codons. They have a TAT bias. And so we thought if DNA repair genes have a TAT codon usage bias, then maybe in the delta TGT strain, where there is no chiosine, since TAT codons are better decoded, maybe you have more of these DNA repair genes. So maybe those strains would be more resistant to DNA damaging agents. So we tested that using UV resistance. So UV, ultraviolet rays damage DNA, and so the cells die. And so at 60 joules here, you have the wild type, and the delta TGT was 4 to 9 times more resistant to this dose of UV than the wild type, which was consistent with the cells having more DNA repair genes. We also looked at it in E. coli, but in E. coli, these genes do not have the same codon usage. They are not more enriched in TAT, and you don't have this UV resistance increased in delta TGT. So now our model is that in presence of stress, there are changes in RNA modifications which impact translation and lead to translational reprogramming of proteins with codon biases, which leads to phenotypic adaptation. This can be a layer of regulation on top of transcriptional regulation of genes that is much better described. So so there is an epigenetic or epitranscriptomic, we can call it epitranscriptomic as well, adaptation to antibiotics. This can also be much more generally adaptation to changing conditions. For example, gut versus urine for E. coli, for example, who has different niches, the gut, the urine, or the external environment, gut versus water. So, because, for example, Vibrio cholerae has two niches, the host gut, but and the aquatic environment. It can be a way to regulate stress, transcriptional regulators. When you look at these regulators in the genome, usually they have codon usage biases, not necessarily for tyrosine, but also for others. Ribosomal proteins as well, they have high codon usage biases. So maybe they can also be regulated by other modifications. So that's an interesting thing to study further in the future. And maybe we can think that such tolerance due to transient modifications of tRNA, maybe they can increase the chances of bacteria to acquire genetic mutations, which would make them resistant to antibiotics. So that's it. I would like to thank all the people involved in this work, who are the group members here, and all our collaborators. And most of all, thank you all for your attention. I hope it was a bit interesting for you. Thanks.
Thank you so much, Dr. Zeynep Baharoğlu. If you wish, we can go to the question and answer section prepared by our team and consisting of questions from our participants.
Okay, I stopped the sharing.
Okay, it's okay. I'm directing the first question. Epigenetic studies in bacteria often rely on deactivating methylation-related genes. Instead, could studying the expression of genes involved in DNA methylation help elucidate the pathways involved in this mechanism?
In the mechanism of DNA methylation, you mean?
In the mechanism, the DNA methylation in the mechanism.
Yes, so DNA methylation is quite well described. So you have the DNA restriction modification systems, which are a bit different because it defends the cells from DNA coming from outside, for example, from phages. But you have also the orphan methylases. They are called orphan because there is a methylase, but there is no restriction enzyme, for example. And these sometimes they participate in regulation of the cell cycle, but sometimes we don't know what they do, like the RNA modification. So it's also an interesting thing to study more.
Thank you for your answer. I'm continuing with the next question. Do we know whether the transcriptional change induced by antibiotic stress can later become genetically fixed in the population by natural selection?
Yeah, that's an interesting question. We think it's possible, but we don't have any evidence yet. So, I think if you allow bacteria, for example, with the tRNA modifications, to become more tolerant and better and reply to an antibiotic stress, then they will have more chance. They will divide more and will have more chances to fix some mutations that would occur in their genome and become more resistant. It's an interesting point.
Thank you for your answer. I'm continuing with the next question. This comes from YouTube. In bacteria, does expression change affecting hundreds of genes across the genome is the exception or the rule? Thanks.
Yeah, it's difficult because I presented some results, and I said we find one protein, RS6A, and we use it to say that it's because of this protein that maybe we see the the effect that we see with tobramycin, but I believe that it's much larger than that. RS6A is one protein, but I believe that many other proteins are regulated this way, and I think the sequences of these transcripts have evolved with this bacteria in their niche. For example, the codon usage of a given gene that is very conserved, like REK, for example, in Vibrio cholerae and E. coli are not the same. So I think every bacteria evolve in their niche and adapt to that niche using their thanks to their codon usage, for example, and hundreds of genes are can be regulated this way. If it's an exception or not, I don't know. It has to be studied more in bacteria, at least. It's not very well known.
Thank you for your answer. I'm moving on to the next question. Can bulk or single-cell methods be able to link the heterogeneity of single-cell susceptibility to the population scale response to antibiotics?
Yeah, that's also very interesting. So we did for this study, we did everything in bulk, but yeah, we have to do single cell methods to visualize heterogeneity and to see whether one phenotype conferred by modification, for example, if a subpopulation of bacteria have more modification, if we can visualize that to ask the question whether these cells are more or less sensitive to antibiotics, and we could make that link, it would be really nice. Yeah.
Thank you for your answer. Another question is, while bacteria adapted to different antibiotics often show overlap in their sets of mutated genes, what is important to know about the extent of parallel evolution at the transcriptomic level?
Yeah. So I think antibiotics are some stress for bacteria, but it's not the major stress that they encounter. For example, in nature, the major stress now would be phages or temperature stress. And so I think what we see when we study response to antibiotic stress, um, even if they are different antibiotics from different families, I think those stress responses are integrated in larger stress responses of bacteria. So, for example, what we see usually is that when we try to see what happens with tobramycin or ciprofloxacin, we end up with oxidative stress response. So maybe bacteria use the stress responses that they already have for other stresses to respond to antibiotics. So maybe that's why there are these overlaps that we see.
Thank you for your answer. Another question is, new non-classical methods that can detect modification, like example as phosphorylation in bacterial DNA backbone. How can we control isogenic bacterial population within different heterogeneity among bacteria of new relations between epigenetics and biochemistry?
Okay, so if I understand correctly, how can we detect the different levels of modifications between different bacteria? Maybe that's the question. So actually, the reason why the RNA modifications in bacteria are not so well studied for now was the technical issue that we could not really detect so well the modification levels and modifications. But now there are new techniques that are emerging. There are some techniques based on next generation sequencing and chemical treatments, and now nanopore sequencing, which is booming, and some labs are really experts on these, and there are papers going out every day. So I think the nanopore technology development and the analysis of data and the emergence of specialists will really help this field in the next years.
Thank you for your answer. I'm moving on with the next question. There is a potential importance of genomic expression patterns in shaping antibiotic-resistant profiles. On the other hand, how can we understand of global transcriptomic rewiring induced by resistant mutations?
Yeah, so I think there is a balance between bacterial growth in the absence of antibiotics, and sometimes antibiotic resistance genes have a cost, and this cost is balanced when bacteria encounter antibiotics. So there is a balance between bacteria having these resistance genes and bacteria which have them but grow slowly maybe, and bacteria which don't have it but they die when there's the antibiotic. Maybe populations are mixed and there is a balance between different populations having or not these genes. And sometimes the expression of antibiotic resistance genes are also regulated. They are not always expressed. So that can be a strategy.
Thank you for your answer. Another question is, much information has been gained from recent studies on epigenome modification of the host by pathogens. What difficulties are experienced in transferring these findings from the laboratory to critically ill patients?
Yeah, so that's not my expertise, really, the clinical part. But yes, there are more and more labs studying the effect of bacteria on host modification in the host, so the human cells. So yeah, I don't know. I'm not very specialist on that.
Thank you. The next question is how can we determine the single-cell response to antibiotics while screening different bacteria and molecules having different mechanisms of action?
Yes, in many cases, the stress responses are found in many different bacteria. And so usually what we do is that we have fluorescent reporters of different stress response regulators or stress responses, and in either in time-lapse microscopy or microfluidic devices, we can follow basically fluorescence of reporters. That's what we usually do.
Thank you for your answer. The next question is droplet-based approach. The question comes from the YouTube. The if current sec text is the dependence on populations to achieve a consensus sequence, how can we determine the epigenetic heterogeneity which exists with different cells of isogenic populations?
Yeah, again, this is done usually with either sequencing techniques or nanopore, but few labs do it.
Thank you for your answer. The next question is, droplet-based approach can provide insights into the heteroresistance of bacterial populations. So are there any limitations to these methods?
Yes, so it's a powerful approach because you can have many droplets and you can put many bacteria in every droplet. The limitation would be rare events because, for example, if you study persistence, so this is a subpopulation of bacteria which are supposed to be dormant or they are not resistant, but they don't die when there's the antibiotic treatment. And these populations are quite rare. So, for example, there are 10 to the minus 5 in the total population. So you would need 10 to the 5 droplets, and it won't be statistically significant anymore. But other than that, the droplet-based approach is actually a really cool way to study this, because you can also change the media and follow what happens to the fate of the population for as long as you want. And also the other limitation, of course, is data analysis, because it generates a lot of images and a lot of data. So you need to be able to store these and analyze it.
Thank you for your answer. I'm moving on to the next question. To overcome microbial therapies that efficiency goes rough because of antibiotic resistance, can antibiotic-loaded nanocarriers determine infected cells and act like a phagocytose mechanisms to release suitable drugs? This can be designed for worldwide non-individual treatments.
Yeah, definitely we need new strategies to be able to treat people with antibiotics. So there are many strategies. There is this one. There can be combination of different molecules in order to enhance existing antibiotics action. There's also the phage therapy.
Thank you for your answer. I continue with the next question. Especially in our country, like any other disease treatment, antibiotics are chosen increasingly. Because of that, maybe with this small effect that we make on bacterial genetic variation, we change their resistance to antibiotics. Can we classify bacteria and the relationship between antibiotics and the result of survival time?
Yes, so for the increasing usage of antibiotics, yes, it can. It does select for more resistant bacteria. Unfortunately, there were some studies of which translate experiments done in the lab for persistence, for example, to a more clinical strain of Pseudomonas inside. So these are in vivo experiments. So usually it's in mice, for example. And we see that what we see in the lab happens also in mice. So probably also in humans, but it's not very well known yet.
Thank you for your answer. The next question is, many of bacterial nucleotide-associated proteins do not appear to be homologous to eukaryotic proteins. So could they be suitable targets for new antibiotics?
Sure, but yes, of course. But then we have to find a molecule that targets them and that can enter the bacteria and that does not enter the human cells. So usually it's the problem is that the antibiotic research is expensive, but the benefit for industries and companies is not so high, because it's not the benefit is not so high. So the private companies do not always want to invest on these studies. So that's why the research on antibiotics is a bit not advancing as it could.
Thank you for your answer. Next question is, while the embryo is developing, the tasks are defined while methylation takes place with the GPG method. Can stem cells transform into any tissue because these tasks are not given?
Yeah, so I'm not
And also,
yes.
sorry for interrupting. Can we prevent certain DNA regions from being methylated?
Yes. So I'm not at all an expert on CPG methylation in eukaryotes, but I know discussing with colleagues that, for example, in the brain tissue, only one methylation of one ribosomal RNA determines the cell fate, whether one cell goes at some point of the brain and not to the other part. So I guess in the embryo it must be the same, but I don't know more.
Thank you for your answer. The next question is, during bacterial infection, the pro-inflammatory response can delay bacterial spread and avoid infection. With evolution results, bacteria's effectors allow bacteria to get colonized, changing host cytonic pathways to control their survival periods. This widely affecting cycle can be blocked by the host biochemical reorganized novel treatments.
Yeah, that's an interesting point as well. I think that's a good field that will grow in the future as well.
Thank you so much for these informative answers. Lastly, I want to ask that what advice would you give to the students studying in the scientific fields?
Oh, that's a big question. I think one should really follow their passion, and if you really want to continue in that field, I think you should be persistent, because sometimes it's difficult, and it seems that there are not many chances to get a position and things like that. But I think if it's really your passion, then go ahead.
Thank you for your helpful advices. On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation.
Thank you very much, and thanks for inviting me, and I was very happy to be here today.
I wish you healthy days.
Thank you. You too.